Good afternoon, everybody, and happy Father's Day. This is the first presentation in this series, Brain Talk, Bridging the Gap Between Treatment, Technology, and Research. The goal of the Brain Foundation is to get to FDA-approved treatments beneficial to the autism population. And we fund not only treatment and research studies, but we try to get access to treatments to our community and facilitate clinical trials leading to FDA-approved treatments. As part of this initiative, we will be supporting Dr. Krigsman's clinic in East Bay and also plan to set up a blood draw site for immunological workups for another study by Dr. Junichi to develop biomarkers. We're also working on setting up EEG evaluations and many other similar initiatives. So we will be announcing these through our newsletters. Today, the presentation will be about autism and GI comorbidities, the research, treatment, and the Bay Area outreach. Dr. Krigsman has consulted with thousands of patients with autism and GI comorbidities, and he will be presenting his latest findings in this area, his peer-reviewed publications, and details about his California Bay Area Treatment Center. Dr. Walker has engaged in research and molecular characterization of IBD in autism, and he will be presenting this latest research and his peer-reviewed publications. The treatment study described today is sponsored by the Brain Foundation. Dr. Krigsman is a pediatrician and pediatric gastroenterologist with expertise in the evaluation and treatment of autism-associated gastrointestinal problems. Until recently, he is now licensed and practices in California. His interest in this unique patient population began in 2001, and during the past 17 years, he has treated over 1,900 children from all over the globe suffering from a variety of gastrointestinal problems. His research interest lies in the characterization of unifying the cellular, molecular, and clinical features of ASD-associated IBD, and his original findings appear in numerous peer-reviewed journals. Dr. Walker is a professor of regenerative medicine at the Wake Forest Institute for Regenerative Medicine in Wake Forest School of Medicine, North Carolina. He received his PhD in genetics and developmental biology in West Virginia University and was a postdoctoral fellow in the molecular genetics program at Wake Forest Baptist Health. Prior to joining the Department of Pediatrics faculty in the section on medical genetics, Dr. Walker's group is focused on the application of integrated multiomics approaches using patient biospecimens to understand the molecular characterization for chronic gastrointestinal symptoms in children with ASD, and to understand how chronic GI problems early in childhood may impact neurodevelopment. The objective of this work is to identify clinically relevant biological markers that can be used for diagnosis and/or as therapeutic targets to the benefit of this vulnerable and underserved population. I'd like to welcome Dr. Krigsman and Dr. Walker. Please take it away. I will stop sharing.
Okay. Okay, hello everybody, whether it's good morning or good afternoon or good evening. I want to wish all the fathers out there a happy Father's Day, particularly Dr. Walker, who is giving up his Father's Day afternoon to be here with us. And, you know, Pramila, as you were reading the bios and all the work Dr. Walker and I have done over the years, it's amazing that, you know, Steve and I go back about 18 years now. And we really started out not knowing where this is going to end up. And after many years of working together, of combining clinical medicine with cutting edge bench science, that really is proven to be the only way to make really significant progress in researching these diseases. And it's a great opportunity that I've had to work with Dr. Walker and to work now with the various patient parent groups, Pramila, the Brain Foundation, Moves Mountains, and thank you to them for organizing the conference and for everything else you do. It's too long. The list is too long to mention one at a time. So I wanted to speak really about And I'm going to move a little bit fast because there's so much information. But we're going to do the best we can. And certainly we have a Q&A afterwards and people can ask questions. So it all begins with recognizing the symptoms in children who have autism. If you can't recognize the symptoms of GI disease, then the parent or caregiver can't even request care. Sometimes symptoms can be obvious: bloody diarrhea, vomiting, weight loss, but more often than not, the symptoms of gastrointestinal disease are much more subtle than that. And the first few minutes, I'm going to just go through basic symptom recognition. And even before that, a basic set of facts. So number one, gastrointestinal disease is more frequent in children with autism than those without it, believe it or not. It took about 15 or 16 years of academic back and forth in the literature to establish this. It is now established beyond question, but you will still find some hardcore data people out there who insist that the perfect study has not yet been done. But for those of us who work with these kids, it's clear GI symptoms are more frequent in ASD than without it. It's critical for us, for the parents and for the caregivers, to recognize that the GI symptoms and signs and symptoms are present in order to proceed with an evaluation. No evaluation can occur unless you know what you're dealing with. The difference between a sign and a symptom is a symptom is something that the patient themselves complains of. They say how they are feeling. I have a headache. I have a stomachache. It's completely subjective. A sign is something that an observer can easily see. Measuring a temperature, seeing somebody sweating, measuring a blood pressure, watching somebody vomit, seeing diarrhea. So the signs are easy. The ASD children have a lot of people that are around them all the time. So recognizing the signs are easy, but it's the symptoms, what they feel. And that's where things become tricky. And that's where we've done a lot of work, and we're going to be getting into that. It's critical for parents to be able to distinguish normal from abnormal stools so that they can know what to say is wrong. It's critical for us to realize that, for, again, parents and caregivers to realize that children with ASD struggle with communicating their thoughts and their feelings, and also in communicating their GI symptoms. So, and I'm not even talking about verbal communication, but even nonverbal communication, even putting your hand on your abdomen, which is the universal symbol for abdominal pain. Most of my patients cannot do that. I often say when I speak that I've had patients who've had their had ruptured appendixes, and they never once put their hand on their belly during that entire time. It's important to recognize that GI symptoms, meaning the feeling of not being well, having stomachache, often erupt as behaviors. So an abrupt aggression, abrupt self-injury. But they can also masquerade as behaviors, meaning that the child that paces back and forth for hours on end, those behaviors, in retrospect, are often, not always, but often found to be the manifestation of an underlying GI disorder. It doesn't have to be a GI disorder. Behaviors, extremes of behaviors are not exclusively the domain of the GI world, but clearly that is probably the most common comorbidity, as many of you probably know. And finally, from both the parent caregiver standpoint and also from the physician standpoint, a high index of suspicion is needed when dealing with the question of whether there is GI disease or not. In other words, when in doubt, move ahead with a diagnostic evaluation. Many caregivers, many practitioners and physicians will often misdiagnose an organic problem, meaning something that is actually measurably wrong with these children, and they misdiagnose it often as a functional disorder. Functional disorders mean that there is no measurable abnormality. Usually the patients who I see have been seen previously by at least one a very highly qualified pediatric gastroenterologist, and they give it a diagnosis of irritable bowel syndrome is the most common functional disorder. But just to give you a background what a functional disorder is, was the Rome 4 criteria that took a number of different GI organs and basically came to a consensus opinion of what specific symptoms are needed to call it a functional disorder, meaning that there's nothing organically wrong that we need to look further into, but rather we are going to label it as either a unspecific motility disturbance, visceral hypersensitivity, meaning, what may be manifested as the perception of pain is not really pain, nothing is really wrong, but your nervous system is just more sensitive to stimuli that normally you and I would not be sensitive to. Perhaps it's a response to meaning the physical symptom of diarrhea, for example, may be the response of altered gut microbiota, or the way the brain just processes what you and I would have considered to be normal signaling is processed abnormally in the brain. These are all how medicine views functional disorders. And many, if not most of the patients that come to me have been labeled with this, and we've shown through really endoscopy, imaging, biopsy, and now more recently with Dr. Walker, who will be speaking about molecular biology, that these are not at all functional, but rather these are organic in nature. And the problem with trying to determine if a patient has a functional disorder or an organic disorder is that it depends, it revolves around asking questions and eliciting answers from the patients or their parents. And the symptoms or the questions that involve symptomatology, what the patient feels, of course, that entire area is just not applicable. But at best, you have the parent's interpretation of what the child feels, the autistic child feels. But because of that, the questionnaires that are used, both in clinical medicine and also in academic medicine, to try and quantify and do research on these disorders really go out the window entirely when it comes to autism, because you cannot give a questionnaire that relies on symptoms, self-reported feelings to these patients. So in order to do our treatment study, we had to come up with an entirely new set of questionnaires. And yes, in truth, these have not been statistically validated because that process takes, it would take years. It would take years, and frankly, we haven't got time to wait. Those patients, those docs, and those practitioners who work with these children and the parents have come to recognize that there are certain behaviors that these children have that are strongly indicative of something wrong abdominally or gastrointestinally, and we've chosen to use to accumulate those questions, put them in a questionnaire, and the way that we go about doing this is that we have the study is that you have a patient that comes in with a specific set of symptoms. The workup on the child is the same workup that you would do on any other child, autistic or not. So based upon the signs that the child's presenting with, whether it's vomiting or weight loss or diarrhea or blood in the stool, there is a certain set of tests of non-invasive testing that we do on any patient with a given set of symptoms or signs. And that's exactly what we do with these patients as well. Usually, the series, a panel of non-invasive tests comes back entirely negative, meaning that there is no measure, there's no clear diagnostic picture or pattern that lets me say this child has this diagnosis, and I will therefore treat him or treat her with a specific treatment. At that point, a physician has a choice of either saying, well, if I don't have a test that gives me a diagnosis, therefore I don't know what to diagnose, so therefore either it's functional or I can't help you. Or you can say that perhaps something is wrong and proceed further with your workup. And that's the latter is the path that we've chosen to go on. If the non-invasive diagnostic tests are negative or do not point to a diagnosis, the next step, as it would be with a non-autistic child, is to obtain a intestinal biopsy, because there you can get much more information. And you're reaching further, of course, into the core of the patient, and you're getting the ability to make a tissue diagnosis. So what are the questions that we ask in our treatment study? How do we get a baseline record of their GI status? So we have two separate GI questionnaires. And one questionnaire deals with the actual bowel movement itself, what the child is doing during the bowel movement, and what do the stools look like. So behavior is associated with the process of passing a stool, and also the specific descriptive components of that bowel movement. Bending forward while passing stool, that's a sign of constipation, actually. That means that you can't pass stool with simply regular colonic motility and relaxing the sphincters and of the anus. You have to generate increased abdominal pressure. A lot of children will do very odd behaviors during the bowel movement. Because it's very painful for them. They move around, they contort, they twist, they stomp. Some kids squat on the toilet seat to generate their intra-abdominal pressure that way. This hyperactivity prior to passing a stool, and we ask in the Likert scale, we estimate all these observations. Hyperactivity prior to the stool, being much calmer after passing the stool, and then the qualities of the stool itself: undigested food in the stool, malodorous stool, the types of odor of the stools, and very interesting as well. Many patients will have this rotting odor to the stool. Others have a chemical odor to the stool, and I suspect that the explanation for that is going to turn out being what type of microbiome they have in the stool. Dr. Walker will be speaking to us soon about our nascent interests in that, and what our plans are for the microbiome exploration. We also want to know about the stool frequency, questions 9 and 10. We also want to know how many times how many days of the week a child tried to have bowel movements but was unsuccessful. These are things that any parent who has a child that's been through this is very well aware of, and they're often shocked when we ask them as part of our routine questionnaire. They're just shocked, and they're relieved that there's actually someone or some group that recognizes these as symptoms that are of value in A, making a diagnosis, and B, furthering the research. So these are the questions that we ask in our bowel movement questionnaire. And again, also the famous Bristol stool scale to describe the texture of the stool. And for those of you who are looking for a Father's Day cake, we have a nice assortment of Bristol stool Father's Day cakes, which hopefully Pramila will be able to serve after the webinar. And then we have a second set of questionnaires. We have a second set of questions in a questionnaire that discusses not the bowel movement or the act of having a stool or the descriptive components of the stool, but rather the externalizing behaviors in general, not around the bowel movement, but behaviors that children have that are often attributed to their autism. Those are called externalizing behaviors, like aggression, for example. We'll see the questions in a moment that we ask about that. And we've learned over the years that aggression, self-injury, hyperactivity, destruction of property can be manifestations of really almost anything that bothers a child. It could be noise, it could be light, but it can also be abdominal pain, especially when you have these extremes of externalizing behaviors that are happening together at the same time with abnormal stools. One of the types of questions that we ask about the externalizing behaviors are posturing, where a child tries to put pressure on their abdomen, in ways that are perhaps unusual, but that these children have learned how to make themselves feel better. Begging on their chest, tapping on their chest, could be an esophageal disorder, reflux disease, arching their back, trying to make yourself gag. That's something that we've learned also is a common disorder because of nausea. They can't vomit on their own. They'll try and make themselves vomit to give themselves relief. The frequency by which parents are called to pick their kids up at school. If you have an ASD specific school where all the kids in the class are ASD and all the people, therapists, teachers there are trained to work with ASD kids, if you have a school like that, and there are so many of them, as you know, and they have to call a parent because that child is unmanageable, then that's something that's extreme. And that needs looking into. Poor appetite in general, food refusal, weight loss, easy fatigue, poor sleep, increased meaning poor sleep, meaning interrupted sleep, waking up at night, or increased sleep latency problems, falling asleep, unusual sleep positions. These are all things, and the photographs on the right will show that. These are all things we've learned over the years to ask, and that they're often manifestations of GI disease. Also, posturing, question 15, the humping into a pillow or blanket, abdominal distension, vomiting, retching, where they make the motions of vomiting, but nothing comes up, rumination, where food comes up into their mouth and then it goes down again. They'll swish it in their mouth, they swallow it, they often play with it before they swallow it again. And again, these are not behaviors, they are more often than not GI symptoms, and they really demand a GI evaluation. Head banging, self-injury, as you see in this photograph over here, severe head banging, perhaps my most severe case, pulling out hair of their head. I should mention also that the pictures I'm showing are extremes of self-injury, extremes of behavior. And many parents, I've been criticized, and appropriately so, for showing these pictures, because parents think, oh, if my child doesn't have these extremes, then they probably don't have the GI symptoms. But I really have to want to emphasize that you don't need to have extremes of self-injury, totally head bruising or your hair half pulled out of your head. It doesn't need that. Even minor amounts of that could be equally as telling. The fact that it gets extreme just means that it's going on for a longer period of time, or that it's much more intense. But by no means should anyone that sees these pictures think, oh, if my child doesn't have a banged up head, then his head banging is probably not something that's organic. That's not the case at all. And these, again, are perhaps more extreme cases of self-injury that you see over here: aggression, scratching, biting, head-butting. These are all the questions that we ask on our autism unique questionnaire. Because if you ask the question specifically, does your child have pain? The answer that the parent may say may be no. But when you ask them these questions, then you have all of a sudden a lot of number fives on the scale. Temper tantrums, intense temper tantrums, aggression, violence, injury to others, and this would be an example of irritability. And this is a child who's crying, and he would do this multiple times per day, no known reason. And we know that this was abdominal pain because he subsequently underwent an endoscopy and a biopsy and was found that he had a treatable inflammatory bowel disease. He only at the very end puts his hand even somewhere near his abdomen. So you can't even rely on that. And then you have another young child who's very quick episode. Okay, also, this child, the second picture, the second video is many, many years, like 15 years ago. This child is growing up to have Crohn's disease. That became clearly apparent only in the last 2 or 3 years. And he needs some strong immune suppressive medications. But this was the very beginning of it. back when he was simply 3 years old. So those are the questions that we ask from a GI standpoint. This is just a quick list. Those symptoms are non-specific. These are just some examples of diagnoses that could cause any of those types of behaviors, any of those signs, or any of those symptoms. So you have to approach these children with an open mind, saying any of these diagnoses can be possible, and do your workup tailored to identify any of these. Because of the nature of autism and the nature of what they end up having, most of the kids with these symptoms will have a negative non-invasive workup and will ultimately need a diagnostic upper endoscopy and colonoscopy. And in the case of autism, I almost always do a capsule endoscopy, or the pill cam, which many of you may have heard of. And the pill cam enables me to view the 2/3 of the intestine, of the small intestine, 2/3 of the gastrointestinal tract that is beyond the reach of the scope. So when you do an upper endoscopy and a colonoscopy and the capsule endoscopy, the pill cam, you get a full view of the GI tract. And when you do that, what you find is that the most common diagnoses by far are the ones in bold: autism-associated enteritis, small bowel inflammation, and an autism-associated colitis, inflammation of the colon. But of course, any of the other diagnoses are possible as well, especially the eosinophilic esophagitis that we see here on top. What is ASD-associated endocolitis? It is, very simply put, it's inflammation of the gastrointestinal tract in patients with autism or somewhere on the spectrum. And this inflammation has unique cellular and molecular characteristics that allow us to distinguish it from other forms of IBD, such as Crohn's disease and ulcerative colitis. So it's a unique inflammation that occurs only in the developmental delay that we call autism. You don't see it in other developmental delays. You don't see it in Down syndrome. You don't see it in mental retardation. You don't see it in genetic syndromes. You don't see this bowel inflammation in neonatal asphyxia. You don't see it in stroke victims, pediatric stroke victims. You see it only in children with the specific deficiencies of development that allow us to call their disease autism. And that means, of course, that the brain disorder, the developmental delay that's occurring in these children is somehow related to this unique bowel inflammation. And that concept is, of course, fascinating, and it speaks to the gut-brain interaction and how that may work. And it brings into question whole questions in areas of microbiome, et cetera. The important part that the parents want to know is, what do you do about this? So when you have patients with these kinds of symptoms who end up having any diagnosis, but in particular the autism-associated endocolitis, what do you do about it? Well, fortunately, the treatment for that disease is really the same, very similar as other types of inflammatory bowel disease, such as Crohn's disease. And we found that treatment of the inflammation results in significant improvement or even complete resolution of the GI symptoms. We also found over the years, we observed over the years, that aside from improvement or resolution of GI symptoms, you also have improvement, a quantifiable improvement in cognition and behavior. And we'll discuss that in just a minute. And that's what we're looking at now. In other words, not only did we find that the GI symptoms improve, which of course you expect when you treat a GI disease with drugs that are designed to treat GI inflammation, but what was very surprising years ago was the observation that their behavior improved as well. And even that's not so surprising, because if you're in pain, we can understand, all of us can, that you can have extremes of behavior. But what was really fascinating is that even in areas of cognition, receptive language, understanding, verbal ability, communication, verbal and nonverbal, those all tend to improve in a quantifiable fashion when we treat the GI disease and see improvement in GI symptoms. Just briefly, the way the study is designed is that we get a baseline. We do the baseline GI surveys on the bottom two rows, the bowel movement chart, as I showed you, and the bowel and the externalizing behaviors. And then at baseline also, we do three separate measures of ASD deficits. And many of you have heard of them, the Violin, the SRS2, and the ABC. And so we get these at baseline. During the 1-year follow-up period, specifically for patients with get a diagnosis of autism-associated endocolitis and are treated for that with medications that are both immunomodulating and immune suppressive and anti-inflammatory. Every week for the next year, the patients fill out those same online questionnaires that you saw. And then at 3 months, we do the ABC. 6 months, we do the SRS2 and the ABC. 9 months, the ABC. Then 12 months, finally at the end, we do all three of them again. Also, during the study, we get baseline molecular characterization. So that's at time of endoscopy and biopsy and diagnosis. We do a characterization of their molecular abnormalities, such as gene transcripts and other biomarkers. And then once the year is done, we get another measurement of those same molecular markers to see if they've changed over the years while we've treated the underlying GI disease. Our preliminary data really is quite fascinating. And I'm sorry we cannot present it to you in graph form, but I wanted to wait to present the graphs until it's a more suitable academic audience. What we found so far is that, as expected, treatment of ASD-associated IBD does result in improvements and often, more often than not, resolution of their GI symptoms. So the diarrhea, the pain, and the stools normalize. The behavioral symptoms, extremes of behavior, the aggression, the self-injury, the pulling out their hair, being unmanageable, being unable to take them outdoors to a mall because you couldn't anticipate what they were going to do. Those all improved markedly or disappeared entirely. It's really quite remarkable. And we also saw in correlation with the improvement in their behaviors, and GI symptoms also came quantifiable cognitive improvement. This is not a cure for autism and should not be interpreted as such, but it does show that there is a measurable improvement in their cognitive ability to the point where patients who were non-communicative became communicative, either verbally or non-verbally. They were able to express to their parents what they were thinking and feeling, which is what a parent wants from their children. And we also found in general that immunomodulation, immunosuppression achieves a superior GI and behavioral cognitive result as compared to anti-inflammatory medications alone. So patients do better GI-wise and behaviorally cognitively when you actually immunomodulate or immune suppress them to some degree as compared to just giving an anti-inflammatory. That really speaks to the significance of the inflammatory process here and how important that is in the overall mechanism of autism. And my belief is that it's not because, I mean, in part, it may be because something from the inflamed GI tract, some molecule is then transmitted to the brain. But it's also very likely that the same inflammation systemically that's attacking the bowel is also attacking the brain. When we give an immune suppressant and modulator, you see improvements in the brain because you are also at the same time as you're decreasing the inflamed intestinal mucosa, you also are, no question about it, having those same immune suppressive medications going to the brain. And finally, I'd like to announce the opening of our of our new office in California, in Pleasanton, California, with a diagnostic surgical center in Fremont, California. I want to thank Pramila and the other parents who prodded me for years to come do this to the West Coast, and who convinced me over perhaps a too long period of time, when the fault is mine, not theirs, that there are many, many patients who just simply cannot travel to a location where I was at that was further away. And I'm more than happy to come to you and to provide whatever help I can in making your lives and your children's lives much more manageable and much more productive and much more meaningful. To schedule an appointment, it's all here. Go to the new patient page at our website. Deb Hunter, you will find her to be a phenomenal, magnificent inpatient, new patient coordinator, and she will walk you through the process of getting registered, enrolled, and seen really within weeks. So it's not going to be a 6-month waiting list or a 1-year waiting list. If the patient is in need, we will get to that patient in a matter of weeks. Thank you very much. I want to turn it over now to Dr. Walker to tell you what the really smart people do with the biopsies that we obtain. Thank you very much.
Just one thing, please ask your questions on the Q&A, and following Dr. Walker's talk, we will get them answered. Thank you.
Okay, are we ready to start?
Yes.
Oh, okay. All right. Well, thank you all for joining. I'm going to follow up with Dr. Krigsman's talk about sort of the background and what it is we're investigating, and to tell you some of the studies that we're doing actually in this patient population and with the samples that we get from the procedures. So Dr. Krigsman and I started collaborating, it's been 17 years ago or something now, but at the time, he had postulated that this IBD-like condition in kids with autism was a unique variant of IBD. So it was not exactly Crohn's disease, it wasn't ulcerative colitis. It had features of both, but it was different. And so coming from the molecular sort of background, I thought, well, how are we going to investigate this? And one of the early studies that I looked at was trying to evaluate, because Crohn's and ulcerative colitis, the known and characterized IBDs, are also quite similar clinically. There sometimes is an issue with determining whether a patient has Crohn's or ulcerative colitis. And the reason it's important to know which they have is because there are some features of those two conditions that are distinct, and it would inform then which treatment strategy you would use. So it's important to know that. And so sometimes they had a patient that they were unable to, they knew they had IBD, but they were unable to determine whether it was Crohn's or ulcerative colitis. And so what did they do? Well, they did a molecular study. So they took biopsies from patients with gene expression profile, and they compared the two. So what's the gene expression profile in Crohn's? And what's the gene expression profile in ulcerative colitis? And what's the overlap? And they found 7 transcripts that distinguish between the two. So if a gene was upregulated in a patient with Crohn's, it was downregulated in the patient with ulcerative colitis. And so there was a panel of those 7. And so I thought, well, you know what we should do is just add a third group. So we can get a group of pediatric children with Crohn's disease, with ulcerative colitis, and then with what we were calling ASD enterocolitis, and compare the molecular profile. And so that's exactly what we did. So our original study was to develop the long goal was to develop a blood-based biomarker, but the initial goal was to just I guess, prove to the pediatric gastroenterology community that the thing that Dr. Krigsman and others have been saying all along, that this is a unique, distinct IBD variant in kids with autism. And so, let me get rid of this. And so the goal of our original early studies were to describe autism, ASD, IBD at the molecular level, and to develop a sensitive and specific tool with the longer term goal of identifying a blood-based biomarker, so that in some cases, if you have a child that meets all the criteria for a colonoscopy, ileocolonoscopy, potentially you could do a blood test, and that blood test would give you an idea what you would find. In other words, would you find inflammation if you went ahead and did the diagnostic ileocolonoscopy, and maybe save the patient and the parent and the clinician from having to go through that. And then finally, the third goal was to provide the necessary evidence-based framework for a personalized treatment approach. Because again, 15 years ago and even up until today, it's not universally appreciated that kids with autism have a unique IBD. And so, as Dr. Krigsman alluded to, oftentimes it's missed, the actual IBD component of a child presenting with GI symptoms is missed. And so the window to give effective treatment is also missed. So, so the study design for the original study was 4 groups of children. So we had children labeled ASD-GI. So those are children with autism, and we had determined they have GI inflammation. And then we also have a bank and a patient population of typically developing children from the GI clinic here at Wake Forest. And we got children with Crohn's disease, ulcerative colitis, and then what we're calling controls here. And in this context, this doesn't mean children that don't have any GI symptoms. It means children have GI symptoms and who are scoped, but who the pathology report shows no abnormality. So those are our controls. And so we took 2 tissues from each of these 60 individuals, one from the terminal ileum, one from the somewhere in the colon. And we published this in a journal called PLOS ONE, and where we had identified a unique gene expression profile in children with ASD. So we found what we thought we would find. There was a panel of about 170 genes that were uniquely differentially expressed in children with autism, no matter if you looked in the TI or elsewhere in the colon, compared to all the other groups. And so the follow-up study, what we really want to get to is a blood-based biomarker. So in order to do that, the next study, we enrolled children with ASD and GI inflammation, and typically developing children with GI symptoms. And in this case, we got a biopsy sample and a blood sample. So there were 4 groups. The children with ASD, we got a biopsy from the terminal ileum that had been indicated on the path report that it was inflamed. Blood samples from the same 12 children, and then another biopsy from somewhere in the colon that was inflamed, and blood samples as well from those same 12. And then we had the comparison group in the typically developing non-inflamed TI and colon, and also blood. And when we compared just the biopsy data from the first study, remember that was a more extensive study, and then the second study that just included kids with autism and controls, none of the Crohn's or UC. We found an overlap now of about 59 transcripts that no matter which study you looked at and where you looked at in the GI tract, 59 transcripts that were differentially expressed and unique to ASD GI children. And then when we took it a step further and looked at those 59 transcripts and compared those to the differential expression in blood, we found a total of 9 genes that actually were differentially expressed in GI tissue and also in blood from children, uniquely in children with autism and GI disorder. And so we published that as a putative blood-based biomarker for ASD-associated ileocolitis. And we now have funding and a study where we're validating this biomarker. It's likely going to be less than 9 transcripts, but if it pans out, we should have a gene expression blood-based biomarker that will give you an indication of whether the child has ASD-associated inflammation in the gut. Okay, so we've been doing those studies for years and continue to do those, but we're now moving in other directions because we have a vast tissue bank and a vast resource here. I'm in a regenerative medicine institute, and so we have lots and lots of things that we can do, and technologies and so on that we can leverage here. So what we're moving towards now is developing an overall conceptual framework to understand dysbiosis. And you've heard a lot about dysbiosis, both in the literature and in the news. And dysbiosis just refers to a non-homeostatic microbiome status in the gut in this instance. And so why are we interested in the microbiome? Well, you probably are aware that there's a bidirectional gut-brain axis. And so things that are generated in the gastrointestinal tract can be transported via the peripheral blood to the brain, and often can enter the brain and have impact there. And then things that leave the brain can travel through peripheral circulation to the gut and impact the gut. So it's a bidirectional brain-gut axis. And this is a little bit more of a detailed study, but I mean, basically what you're looking at, what we can look at, we're not looking at brain tissue, so we're only looking at what's made in the gut, and then we can postulate what that's doing in terms of in the brain. And one of the reasons this is really interesting, and I don't know if Dr. Krigsman pointed this out in great detail, but one of the fascinating things about this ASD-GI disorder is the early age of onset. So when we did our original PLOS study, the average age of those 30 children in that study was about 5 years old. And the average age of the children in all three of the other groups, all typically developing children, Crohn's disease, UC, and control, the average age was about puberty, 12 to 13 years old. So that argues that this GI disorder in children with autism, the onset is at a much earlier age, and then that lends itself to all sorts of interesting ideas. You know, the chicken and the egg. Does having autism or developmental delay predispose you to chronic GI symptoms, or the other way around? Does having chronic GI symptoms, particularly at an early age during a critical developmental window, does that impact your neurodevelopment? So in other words, if you could stop that in its tracks, like doing the things that Dr. Krigsman does, could you actually stop a child from actually ever getting a developmental diagnosis? Interesting to postulate. Okay, so the research strategies that you may be aware of to look at microbiome is one of the standard ways to look at it is you have a control group and a patient group, you collect stool samples, and I'll say that microbiome typically, so microbiome refers to all the microbiota. In this case, we're talking about the gut, and so that's the bacteria, fungi, and viruses. Most of the studies, I'd say the majority of studies published right now, first of all, use stool as a proxy for what's going on in the gut. And secondly, they do what's called 16S sequencing, which is specific for bacteria. So you're not catching viruses and any other thing besides bacteria. There's a second technology called metagenomic sequencing. And metagenomic sequencing is actually sequencing everything in the sample, and then lining up the sequencing data to the database sequence that's available. And by that, doing that, you can remove the human-based sequences, and what you're left with is what's bacteria, virus, and fungi. So when you have that information, you can look at metagenomics, and you can look at the fecal microbiota composition. And what we're typically looking for is the biome in the gut diverse, and are there what they're calling undesirable species, are they overpopulating the gut, and so on. Second type of study you can do is using biome from humans and transplanting that into mouse and seeing what the changes are. We don't do any of that. The third type of study that you probably are aware of if you're following the field at all is this microbiota transfer therapy. And so the group at Arizona under Jim Adams has been doing that now and has published that, and did a study in children where they took GI symptomatic children. In this case, they weren't I'm going to say the hard cases that necessarily Dr. Krigsman sees, but it was children with autism who have GI symptoms. And also, obviously, they meet a threshold for the autism diagnosis. And when they essentially remove their entire gut biome and then replace it with biome from a pooled healthy donor source, and then they evaluate changes in GI symptoms and in characteristics of autism, and find that the children, for the most part, improved in all domains, in some cases dramatically. And interestingly, in a 2-year follow-up, these changes were maintained. And so that's really exciting. It's not to say that's going to cure everybody, but it's really an interesting first step. We're now partnering with another investigator there on the West Coast who also does FNT, but she does it. This is Dr. Spinazan, and she does this type of therapy, but we'll call it familial FNT, meaning instead of using a pooled donor source, healthy donor source for the microbiome that you transplant, she gets the material from a family member, first-degree family member. So it's an interesting concept, and we're looking at that. I'm actually working with Dr. Adams' group to analyze samples that they collected during the clinical trial to understand the gene expression changes and also the metabolite changes that occur with FNT. And then finally, I'm sure you're aware, a dietary intervention is another way to impact, probiotic and prebiotic therapy to impact the microbiome. And so the approaches to study, basically, we take samples from within the gut, and what we're looking at, we'd like to look at metabolites, obviously the microbe profiling, and then genes and proteins. And the question is, putting all that information together and understanding it might give us a more clear understanding of how the microbes are interacting with the host, because the microbes, after all, are making a tremendous number of metabolites that impact metabolic processes within the host. And so if those are symbiotic and healthy, then that's a good thing. If they're detrimental, then that's a bad thing. So understanding that is important. And getting back to Jim Adams' study for a moment, they know, and this is true with a lot of medical therapy I'm finding, they know that it works pretty well for most people, but understanding what the mechanism is, that's the challenging part. And the reason that's useful, it's useful for a couple of reasons, but one of the reasons is if you can identify which part of the microbiome is responsible for the improvement, for example, could be you don't have to go through an entire FMT transfer. You could theoretically then do a more targeted approach. But anyway, that's down the road a little bit. What we're trying to do is understand mechanism here. So there have been, in the last couple of years, lots of systematic reviews, meta-analyses, and so forth of all the microbiome data that's been published on children with autism. And in this study from 2019, they looked at the differences in microbiota composition in children with ASD compared to their unaffected SIVs and healthy controls. And they chose 28 studies that met the criteria. And the majority of the studies included fecal samples. So again, I'm going to get to that in just a minute, why I think that's a limitation, but that's typically what people do because it's a sample that's not difficult to get. And a couple of the studies in this meta-analysis also looked at mucosal biopsies from different sites within the gut. And what they found was there's a heterogeneity in the ASD diagnostic methodology in the site of the gut that's sampled, if they sampled the gut site, and the laboratory methods used. And so they say all these variables made a meta-analysis inappropriate. And so the take-home message was the gut microbiota is altered in ASD, although a lot more work is needed. And so then there was a second review of all the published work that was published last year. And in this case, they point out the GI distress, they use distress, is present in up to 90% of the ASD cases. The prevalence number, by the way, and Dr. Krigsman alluded to this, the prevalence number is all over the place, and it has to do a lot with how we define gastrointestinal distress, gastrointestinal symptoms, gastrointestinal disease, depends what you call it and what you're talking about. And we use different words. Sometimes we're talking about the same thing, sometimes we're not. So that's an issue. In the second, so they looked at 19 studies that met their inclusion criteria. And what they found was that research continued to report differences between the microbiota of kids with individuals with autism. However, the type and abundance of bacteria that within different cohorts, if you will, or even within different individuals is not always the same. In other words, we don't know what constitutes what we would call an ASD dysbiosis. And so there are some commonalities, but there by no means is there. And in fact, one of the reasons for that is we don't know what constitutes necessarily. There isn't a set healthy biome either. We know what a healthy biome looks like, but they don't all look the same is what I'm getting at. So, and they would argue, they concluded that rigorous methodologies are required to support and strengthen the reliability of what's being reported right now. And they pointed to the need for subgrouping, which is something that we've recognized from the beginning. And it's really important that so the way we subgroup is we don't look at all children with autism. We look at children with autism who have chronic GI disorder. And so that's a subgroup within the ASD group. So we're not solving autism. We're trying to solve or understand how kids with autism, why they're more predisposed, it seems, to a chronic gastrointestinal disorder. And by doing this subgrouping, so getting groups of individuals who are more similar to each other than different, when you find differences then between that and the control group, it's more meaningful and more widely applicable. So again, back to the approaches, the standard approach to look at microbiome as a control group and a patient group, you get a stool sample and then you do some type of analysis. A pilot study that we ran and we just completed the data for not that long ago is the question we're trying to ask is the fecal microbiome a good proxy for the mucosal microbiome? And we believe it isn't. And what does that actually mean? We think probably if you sampled the microbiome locally in different anatomic locations within the gut, you would find a different composition. So mucosal microbiome composition will vary by anatomic location. It will differ significantly from the fecal microbiome because remember, the fecal microbiome is a compilation of everything in the gut, theoretically. And then a third hypothesis was that the microbiome will differ significantly between children with autism and typically developing children. So for this little pilot study, we get biopsies when Dr. Krigsman does a procedure, we get biopsies from multiple locations within the upper and lower GI tract. So for this study, we took samples from 5 symptomatic male children who either have ASD or developmental delay who are undergoing endoscopy. And we assayed tissues from the ileum, the transverse colon. So these are disparate locations, the terminal ileum, the ileum, the transverse colon, and the rectum. And so we extracted microbial DNA and RNA from the biopsy samples. We performed what I said before was this shotgun metagenomic sequencing, which sequences everything. You analyze the sequence reads to reference databases, and you remove all the human-based sequence information. What you're left with is bacteria, fungi, and viruses. And then you identify and quantify these organisms. And for this case, we're looking at bacteria. And you get images that look like this. And so across the top are patient 1 through 5, and then there are 3 columns for each of the patients. And if you look down at the bottom, the first column for each patient represents the microbiome, bacterial microbiome in the terminal ileum, the transverse colon, and the rectum. And without really necessarily knowing what you're looking at, this is a measure of microbial diversity. And I think you can appreciate from looking at this that, and this is a summary of what you look at in the terminal ileum across an average across all 5 patients, the transverse colon across all 5 patients, and then the rectum. And so we're going to publish this, so you'll have all the details when we do that. But for right now, what I'll tell you is microbial diversity varied between the 5 individuals, which we would expect. That would be any 5 individuals, you see the same thing. Diversity varied between anatomic locations. We would have expected that, we hypothesized that, but it had not been definitively shown before, but it has been now. And then finally, the microbial composition of the TI was less diverse, more similar across the 5 individuals compared to the transverse colon and the rectum. So we have long suspected that the terminal ileum is a hotspot, if you will, for in these children with ASD-associated enterocolitis. So we're following up with these studies. And so I wanted to point out, Dr. Krigsman alluded to this, but didn't really, I'm going to give you a few numbers. We have a biorepository that we've been collecting over the last many years. It's under IRB approval. And so we have samples from children with autism, 1,300 children with single encounters, means he saw him at least one time. We have another 150 for which they've been to see him 2 or more times. We got a full sample set. And then 20 with 3 encounters and 11 with 4 encounters. And then we have samples from 400, now almost 500 typically developing control children. And the samples in this biorepository are pretty extensive. So for each child with autism, we have 12 to 16 GI biopsies that are in a preservative. We have another 2 to 4 flash frozen biopsies from the same patient. We have whole blood and Paxton tubes that we can do gene expression analysis. We have serum and we have plasma. And for the control children, we have comparable data sets. So we are a sample set so that we can do an awful lot of molecular work with samples like this. And I'll go one more minute or two. The next thing we're looking at is to create an in vitro model based on human-based tissue. So you can do this a number of ways. You can do intestinal biopsy, tissue resection. You can use pluripotent stem cells. And you can look at multi-omic approaches to identify what's going on in these tissues. We use an approach that's pretty ubiquitous here at the Institute called making organoids. And how you make an organoid, basically you take a cell source, and in this case it's going to be biopsies from kids with autism. So when Dr. Krigsman takes a biopsy, he'll send it to us in media overnight. We can isolate the crypts which contain the pluripotent cells, and they will then grow, if we're lucky, and we have been so far, and to create a mini gut, if you will. And this mini gut will theoretically correspond to features of the person from which they come. So we can make mini guts from kids with autism and mini guts from controls, and then compare characteristics of those. And we got the person who's working with this with me, we have a mini gut organoid made from an autism GI patient. And he argues that these organoids, morphologically at least, we haven't done the molecular work yet, but look differently than the healthy control, which you see in the third panel. So with that, I'll end, and the collaborators, Dr. Krigsman, Dr. Reeves here at Wake Forest does the metagenomic work for me. Dr. Bishop helps with the gut organoid and model development work in our organoid facility. That's it. Thank you so much.
I'd call upon Dr. Krigsman to now answer questions on the Q&A.
Okay. Okay, my video is on?
Yes.
Can you all see me? Great, okay. Okay, so we have a number of questions. I'd like to address many of them, as many as we can. So one question is sadly, it's a very interesting one. Is there any way to diagnose someone, an adult, who is homebound, unable to be transported for examination and treatment? And that's that really is, the answer is really no. It's probably not. Well, it's unlikely to be able to diagnose him or her. However, we can certainly, there are certainly tests that we can do that are non-invasive tests that can be done. There are stool tests or blood tests. We can do a portable abdominal x-ray. There are things that we can do that can give us a good idea of whether there's something that's treatable or not. So although this person may, in fact, if they cannot be transported, in fact, they're so ill or so out of control that they just cannot be homebound, there are still things that we can do that can give us some idea and hopefully an idea of how to treat the problem once we identify it. And remember, the problems could be many. There are so many different diagnoses that we've come across while evaluating these children. So, and sometimes in a patient like this, if you can't do a full diagnostic workup and you really don't have a diagnosis because of the patient's condition, sometimes it's okay to treat empirically, meaning without knowing the diagnosis. If you've done due diligence and you simply cannot get all the answers to all the questions you have beforehand, there are cases, there are circumstances that would allow for that kind of empiric treatment. And maybe if I strongly suspect that it's acid reflux, even without proof, so we can give long-term acid blockage, for example. And there's many other things that we could do empirically, bearing in mind that we really don't know the diagnosis. Another question, are we following the Arizona State research on using MTT to treat ASD? So again, what I do as a gastroenterologist is I treat GI disease. Now, I'm interested in the GI disease of ASD. Initially, my interest was to treat the GI disease to get rid of their GI disease, and then we've stumbled across is that it happens to improve autism also. And perhaps when our treatment study is done, we will be able to emphatically state that treating GI disease is one of the ways we can help treat autism. But it certainly would not be the way to treat autism, meaning if there is no GI disease, then you would not use medications for GI disease to treat autism. So it's a very important distinction to make, is that what I'm interested in and what I do is identifying GI disease in these children and treating it for the purpose of treating their GI disease. At least that's what we can say at the present time. One person asked if there is a link to do the self-assessment, the questionnaire that we do, and I don't have that link. I don't distribute that because right now we have not validated the questions, as I mentioned before. Now, the study itself will be on some level self-validating. In other words, if we find that identifying a specific GI disease and treating it and these specific behaviors go away, then that in itself is a validation of sorts of the question, of the questionnaire. Until that's done, I'm reluctant to distribute it as a questionnaire for parents to do. And also bear in mind that the questionnaire is only a questionnaire that would allow parents to determine that their child has GI symptoms. If hearing the lecture, any of those things rang a bell, any of those behaviors, and also if you can really watch any of my lectures that are available online through the Brain Foundation and other sources, and I go over all of the symptoms, perhaps not in questionnaire form, but they're all there. And you certainly could use that to make your own assessment. But does my child need to have a GI evaluation? Well, if any of those issues are issues, behaviorally or from a stool descriptive standpoint, then yes, they do need a GI evaluation for some type of a diagnosis. Another person asks, the immunomodulation that I discussed that we very often use in treating the ASD-associated endocolitis, is it IVIG or a subcutaneous IG? Well, actually, it's neither. IV immune gamma-globulin or subcutaneous gamma-globulin are not established or known treatments for inflammatory bowel disease. And because what these children most often has is an inflammatory bowel disease, then I am confined to using just those medications that are approved for inflammatory bowel disease, and that does not include IVIG. Having said that, just my experience, I have a number of patients who are getting IVIG for other diagnoses of immune deficiencies that they have. And it's clear that there are certainly a subset of patients whose GI symptoms do get better when they get their IVIG. So those patients who, for example, get it on a monthly basis, we at times find that for the week or so after the IVIG infusion, there's an improvement in their GI symptoms, and then things go downhill again until the next infusion. But certainly we don't use IVIG for the GI diagnoses. The question was asked, in our treatment study, the way that we're doing it, how is the design valid without a control group? Meaning we're taking all patients who have a confirmed diagnosis of autism-associated inflammatory bowel disease and treating them and seeing how they respond. It is true, there is no control group. There is no group of patients that we perform endoscopy on, find a disease on, and then don't treat. So I think the answer to the question is self-evident. No parent would ever allow their child to undergo general anesthesia, endoscopy, biopsy, and then risk being placed in a control group, a blinded control group, where they may get a placebo. Not when there's clear evidence accumulated over the years that clearly shows that patients do better when they're treated with some sort of IBD medication. So you will not get a control group. Now, perhaps at some point we'll be able to do a study that compares one treatment to another. For example, one study arm will have FMT or MMT as the first treatment, whereas the second arm will have an anti-inflammatory and immunomodulator as the first arm, and then we'll have a crossover at some point. That's something that we could do in the future. But to have a placebo control group, you're never going to get, and it's really unethical even to do that, since we know that so many patients do better with treatment. A number of people asked if I see patients over the age of 19. I do. I see them well into their 20s. I even have some patients in their 30s. That would be more on a case-by-case basis. And the best thing would be if you have an older patient, someone in their late 20s or 30s, schedule a phone call or a Skype video, and we'll discuss the specifics, and we'll come to some sort of a decision if that kind of patient, if there are other comorbidities, what the specifics are, and we'll deal with it on a case-by-case basis. I see a good friend, Edward. I'm not going to say your last name, but it's good to see your name up here, Ed. The question is that there's, for the medications that are used, which ones are used and how long. So there are a number of medications that we use. I try and have a standard order of medications so as to make the treatment study as defined as possible, meaning that there's less variation from patient to patient as far as what medications they're getting. But initially, almost all patients begin with a course of induction corticosteroids for a period of 45 to 60 days. The reason being that corticosteroids, just over the years, has been found to be the single best drug for a rapid improvement in their GI symptoms. And then after that, I will make every effort to remove steroids entirely, have them on anti-inflammatories, see if we can maintain their remission of their GI symptoms on non-steroidal anti-inflammatories. And if we can't, It depends how severe their relapse is. Some patients will need to go back on a second course of steroids, and every child will take a different road at that point. Sometimes the relapse is so severe that they need an immune modulator, like methotrexate or 6-MP. And then we have a number of patients like we have in Crohn's disease who prove that they need something even more than that. They need biologic agents. And the most common one that I use is adalimumab, or by the brand name Humira. It depends on the patient course, and these will all be looked at very carefully when we get our data together and can analyze it. We'll be able to show how many patients responded to initial steroids, how many needed to go to immune modulation, and it'll be a very, very fascinating analytical part of the study.
I think there were questions on how often you plan to see patients in the Bay Area and insurance and all that.
So, so right now I've made a commitment to come to the Bay Area on a monthly basis, once a month. Initially, at least, I plan on having one day in the outpatient setting, in an office setting, and then one day per month in the endoscopy center for those patients who do end up needing an endoscopy. At least what it looks like is that very quickly we're going to have to double that and make it 2 office days and 2 surgical center days per month, but we'll see how that goes. As far as insurance goes, I do not accept insurance. And there are a variety of reasons for that. The main reason being that the amount of time that it takes to get a thorough history of a patient is not communicative with diseases that are often subtle, in a patient population where the parents are extremely well-read and extremely well-educated. Insurance companies simply don't reimburse anywhere near the amount that that just the business of medicine would demand. Unfortunately, medicine, as many of you know, because of reimbursement rates, it's become a high volume throughput. So you do okay, you stay above water if you see high volume. High volume means very short patient interaction time, and that simply does not work with this disease. That is the main reason. However, the work that I do is all, it is the standard of care, meaning it isn't that we're doing nothing that any other pediatric GI doc wouldn't do, given these same set of symptoms. So for that reason, it's all standard of care. Depending on the details of a particular health insurance plan, it's all reimbursable. And the vast majority of my patients get reimbursed either 80% or 90%, some even 100%. If you're stuck in an HMO that has no out-of-network benefits, we at times will be able to fight for you and advocate for you with your insurance company to extend you out-of-network benefits if you've been seen in-network by a number of at least 1 or 2 gastroenterologists and have not yet arrived at a diagnosis or have not yet had symptomatic improvement. That's a broad answer to a question that can be very detailed, and of course we can answer more specifically when you call the office. It's an important point for me to make that the decision by any parent to have their child evaluated for GI symptoms should not be in any way because of any study that we're doing. The study is something that interested me for many years, but it is not. The study is not the reason to investigate a child with GI symptoms. A child with GI symptoms deserves a full investigation of their symptoms, just like any other child would for those same symptoms, study or not, autism or not. So the decision of any parent to pursue a workup is simply to answer the question, if my child is suffering because of GI symptoms and I can make their life easier and more productive and more manageable with diagnosing what's wrong with them and treating it and relieving them of these symptoms, then that's the reason to do all of it. It's the only reason. The reason is not and cannot be for research purposes. One person asked about JAK inhibitors and if that's used as part of this. And JAK inhibitors could be used. I am very curious to know how my patients with their unique type of bowel inflammation would respond to that. But the JAK inhibitors are relatively new on the scene, and they would need insurance company approval. They're only approved after you've tried a number of other interventions. And I have not yet had a patient who's old enough, who's been through enough alternate medications to be a candidate for approval for JAK inhibitors. Okay, I think, are there any other questions, Camilla, that you see here that you would like to see answered?
There's a question about GI symptoms were present when he was younger, but less severe as he got older. Is it possible for him to benefit now from immunotherapies?
The answer is yes.
There are a number of questions on that vein, like about there is no overt GI symptom today. Should I still investigate?
Okay, so if there are no overt GI symptoms, then there's no justification to do anything more than non-invasive testing. So yeah, if it's always, if the parents are wondering whether there are GI symptoms, it's always worth, in my opinion, a phone call, one consultation. And I'll poke and I'll prod and I'll try and uncover any possible symptom that there may be. But if there isn't any, then the extent of the evaluation will be non-invasive. It'll be blood testing, stool testing, urine testing, and bowel X-ray. And because there's no question that there are children with autism who do not have GI symptoms. What's very common also is what you mentioned just a minute ago, that we have a 17-year-old, for example, who when they were 3, 4, and 5 had all of these symptoms, and even perhaps to the extreme, no proper GI evaluation was done at the time, but over the course of time, their GI symptoms really lessened to the point where they're either minimal now or they don't exist. So those are patients I'm very interested in as well. And I suspect that there probably are ongoing GI symptoms, probably that are being manifested as behaviors as opposed to abnormalities of the stool or of the bowel movement itself. So it's hard to give a clear-cut answer, but just to say that if the parents have a suspicion, use the parents' intuition, the maternal intuition in particular. The intuition tells you that something might be wrong and you want to investigate it further. And certainly, there's no harm to making a phone call and to taking it from there.
Another question that was also interesting to me was, would you consider prescribing pill camera capsule endoscopy without doing a scope if the patient has disease? And this is also relating to the other question about if the patient cannot move around and needs to be diagnosed.
So that's a very interesting question. And a lot of parents do ask that question. So, there are certain criteria that as gastroenterologists we use. And there are certain symptom complexes where the next step is to perform an endoscopy. capsule endoscopy is only very recently was approved to be an initial evaluation for very specific GI symptoms. So to the extent that doing only a capsule endoscopy conforms to standard of care for the specific list of symptoms the patient has, then yes, in that group of patients, it would be appropriate. The practical problem is that because ASD patients, it's so unusual for them to actually give you their symptom, transmit what's wrong with them. You're usually limiting yourself by picking a particular diagnostic study as far as endoscopy and saying, I'm going to do this, but not that. I'll do only an upper endoscopy, but nothing else. I'll do only colonoscopy, but nothing else. I learned many years ago, because years ago I tried to do that. I tried to, to the best of my ability to follow standard of care in patients who talk to you, and only perform an endoscopy of that area of the GI tract that the symptoms seem to point to. And I discovered very quickly that I would often miss out on other areas of the GI tract, that I had to go back and then scope that area too. So I learned quickly that I take the opportunity, they're under anesthesia, get everything done all at once. Look at the entirety of the gastrointestinal tract. Don't leave anything in the dark. Don't guess. And that was something that's it's one of the unique approaches that we have in autism, as opposed to non-ASD patients. So in general, it's not a good idea, but there clearly are some patients who wouldn't meet that criteria and standard of care. And if they do, then nothing is wrong with doing that. Okay,
Anything for
else?
Dr. Walker? Or
yes, this is an interesting question, actually. One person asked, a patient with ongoing diarrhea who had a normal endoscopy, what to do? Is there anything left to do for that child? And that question opens up a big can of worms, okay? Because a normal endoscopy is somewhat subjective, believe it or not. A normal endoscopy, what's considered pathologic, that was the question that got me and Dr. Walker started many years ago. That what some pathologists and some gastroenterologists interpret as normal endoscopy, which in fact is not normal. And we've shown the abnormalities using immunostaining and molecular techniques. So when you say that there's chronic diarrhea, but there's a normal endoscopy, my first question would be, was it in fact normal? I would want to see the endoscopy. I'd want to know if there was a capsule endoscopy done, because if a PillCam was not done, then 2/3 of the gastrointestinal tract has not been visualized. So how do you know that there's no pathology that's treatable that you can see as causing this diarrhea? So that's a great question. It really is a great question. So my answer is yes, there's lots of things left to do for that child.
There was one more question at the end about, and we'll end with that, about C. diff toxin A and B. That's the last question.
Another great question. Okay. So this was something that really, I had no exposure to until really the last 10 years or so. The idea that you can have C. diff toxins A and B being positive in a patient without the chronic diarrhea and not without the classical severe symptoms of C. difficile colitis. So I have seen patients who do that, and to me, I don't know yet what it means. I do not know what it means. I have not noticed any patterns of inflammation that are unique to those kinds of kids. I would just say simply that one of the dictums that I learned years ago from a very wise chairman that I had, a mentor, is that you treat symptoms and disease. You don't treat lab results. So if there's something, if there's a C. diff toxin A and B that's there, but there are no symptoms, there is no diarrhea, there is no pain, there is no growth issues, there's no severe constipation, no abdominal distension, no rumination, no reflux, no nothing. There's no GI symptom there, just a C. difficile toxin A and B. So I, as a gastroenterologist, that practices standard of care would say, I don't know what to make of this. And I wouldn't treat the C. difficile. Now, however, we know that treating IBD, for example, with antibiotics, now that's standard of care. So using particularly antibiotics that are effective against anaerobes are one of the things that we routinely do in patients who have Crohn's disease. And that's the reason why I'll prescribe those antibiotics in patients with the autistic variant of IBD. So it's something we could give that kind of a patient an antibiotic, but not because of a lab abnormality, but because of the possibility of there being an IBD, which might be responsive to an antibiotic.
So with that, I'd like to thank Dr. Krigsman, Dr. Walker. It was a very interesting conversation and lectures, and I hope everybody found it useful. Thank you all for participating today. So hope to see you in California soon, and many of the patients too. Thank you.